Right. Well, thank you again, everyone, for attending our Innovative Discovery Series. I'm Claudine Jurkovits, Director of Clinical Research in the Institute for Research IREACH at Pediatric. And it is my great pleasure today to introduce Dr. Rodney Scott, who will talk about systems-level mechanisms of memory impairment. Before I introduce him, let me mention some housekeeping issue. So we have a few other events coming up. First, we continue extending CME credits for our tech talks and our ID series presentations. We will distribute the information via email to those who registered for today's event. If you did not register prior to today's talk, you can still obtain CME code information from Deborah Rees, and Deborah will include her email into the chat. So on May 7th, we have a tech talk, which will be on a Thursday at noon, from Dr. Kagri Ozdemir, who is a faculty at Delaware State University, and will talk about exploring NIH All-Overs research program. On June 4th, we have another tech talk, which is also on Thursday at noon, and that will be our own Kim Williams and Alex Mapp from Pediatric. We'll talk about survey design, steps to building a research survey. And finally, our next ID series will be on June 23rd, and that's a Tuesday. Dr. Freda Patterson from University of Delaware will give the presentation. We don't have her title yet, but Dr. Patterson is an expert in biobehavioral rhythms of cardiometabolic risk factors and also of sleep and circadian health. So I'm sure it will be really a fascinating presentation. So make sure that it is on your calendar, June 23rd. So again, I'm very excited and honored to introduce Dr. Rodney Scott today. Dr. Scott has multiple titles and functions. He is a division chief of pediatric neurology at Nemours here in Delaware. He's also the director of the Nemours Neuroscience Center that he founded in 2025. And he is professor of pediatrics and neurology at Sidney Kimmel Medical Center at Thomas Jefferson University in Philadelphia, as well as professor of psychological and brain science at University of Delaware and professor of biomedical engineering at University of Delaware. He obtained an MD at University of Zimbabwe and a PhD in neurosciences from University of London. He did his residency in pediatrics and fellowship in pediatric neurology in London hospitals, and has had numerous awards. For example, was named Researcher of the Year at Nemours in 2025. He has been the principal investigator on multiple foundation and NIH grants, notably receiving an R01 Transformative Research Award from the NIH Office of the Director last year, and this is a very prestigious award. He has a long list of manuscripts, wrote several books, and is a sought-after speaker, both nationally and internationally. So I really think we are very lucky to have today Dr. Scott giving us a presentation. Thank you so much, Rod.
Thank you very much, Claudine. After that introduction, I'm scared that I'm going to disappoint you all, but I'll do my best.
I don't think so.
All right, let me just share my screen. I assume it's working, is that right?
Yes, looks good.
Yeah. All right, so thank you very much. I'm excited to come and talk to you all today about learning and memory impairments in neurological disease. And I want to do this or take you through a series of arguments that takes me to why I want to understand this from a systems neuroscience perspective. What I want to talk about today is the burden of neurological disease to convince you that this is something that's worth study. I'll talk about the main brain structure briefly in the beginning and then much more towards the end, which is the hippocampus, which is the main brain structure involved in learning and memory. And then I want to really spend a bit of time philosophically, I guess, talking about how we think about disease in a very classical way, and where that works well and where that fails, and how reconceptualizing the way we think about disease in a complex adaptive systems model actually allows us to have broader concepts and broader approaches to treatment than we currently have. And then I'll show you how we've been trying to take these concepts forward, trying to understand pathophysiology and treatment of learning and memory impairments. Neurological disease has recently taken over from cardiac disease as the most burdensome type of disorder affecting healthy life every year. So there's 443 million years of healthy life lost per year as a function of death and disability from neurological disease, and it costs us about $800 billion per year in the USA. People with neurological disease obviously can present with a very wide range of symptoms. So motor symptoms, sensory symptoms, emotional, behavioral. But the ones I want to concentrate on today are the learning and memory impairments. And these are really important because about 7 to 8% of the entire general population have learning and memory impairments, often associated with disorders such as Alzheimer's disease, epilepsy, autism spectrum disorders, amongst many others. So the structure in the brain that's most involved in learning and memory is the hippocampus. And it's this structure which lives in the medial temporal lobe of the brain and consists of these two C-shaped structures, the dentate gyrus and the cornu eminence. It's part of the limbic system and has a very key role in cognitive and emotional processing. It's absolutely critical for encoding and consolidating memories, as we learned from the procedures that were done on HM. And it also has a very important role in executive functions such as working memory, attention, state shifting, through its projections to another brain structure called the prefrontal cortex. So in learning and memory impairments, we want to understand what it is about disease that's causing impairment at the level of the hippocampus and the prefrontal cortex to some extent, in order to be able to identify and think about treatments which may be able to recover some of the function in the hippocampus. So the way we usually think about disease is in this very classical way, where we think there is a specific cause of the disease. This leads to some biological mechanism, some upregulation or downregulation of a biological process. And it's that defines the disease, and the disease generates symptoms. Where physicians come in is right here. So the physician comes in, identifies a whole bunch of symptoms, tries to relate that to a disease or make a disease diagnosis, and therefore infer something about a biological mechanism and a specific cause. And we do that because we want a treatment which is directly targeted to the specific cause, because then we break that chain of things that I've just shown you and hopefully people get better. This has stood medicine in very good stead for a very long time. This has been a very powerful way of thinking about disease, not only neurological disease, but disease in general. And sometimes it works extraordinarily well. So in this cause-effect treatment type of paradigm, we can think of several different types of scenarios. So the first would be this one. So on the left is the meningococcus, which is the bacteria that causes a particular type of meningitis. What the physician sees in these children are children with fever, seizure, unwellness, all of these symptoms that are here. We come in, we diagnose meningitis, we do a lumbar puncture, find out what the bug is, give the right antibiotic, and hopefully everything gets better. So that's a reasonably linear process like the one that I showed you before. Sometimes we don't do it at the level of cause, but we are able to identify something at the level of mechanism, which still allows us to make a clinical judgment and a treatment judgment on the basis of what we know about a metabolic process. So this is phenylketonuria. This is a metabolic process of that disorder. It's a metabolic disorder in children, which is due to an absence of an enzyme called phenylalanine hydroxylase. What we know is most phenylalanine comes from dietary protein. So if you don't restrict dietary protein, then children get a whole bunch of symptoms: musty smell, changes in skin, nausea, vomiting, growth delay, developmental delay, tremors. What we do know is if you take protein out of the diet, then you can get very positive effects. And in the world of neonatal screening, we identify this disorder very early, institute these low protein diets, and the children do extremely well as a consequence of that. So again, you've got a pretty linear effect. However, let's think a little bit about Alzheimer's disease. So these are amyloid plaques in Alzheimer's disease, and when these build up, you get a whole bunch of symptoms: forgetfulness, sleeping abnormalities, challenging behaviors, loss of memory, etc., etc. And we call that cluster of symptoms Alzheimer's disease or dementia. And we have really good treatments for clearing up amyloid plaques now. However, these drugs, which very much clear up the amyloid plaque, have very limited positive effect. So they slow the rate of progress of the Alzheimer's disease somewhat. They're not having no effect, but it's certainly not a very dramatic effect like you get in the meningitis or the phenylketonuria type of scenarios. So there has to be some limitation to this model. It doesn't work across all diseases. So it's worth thinking about what the limitations are, because then we can think about how we construct an alternative way of thinking about the problem. So these assumptions include things like an assumption of a single isolated cause and mechanism. It is that very linear cause and effect thinking. There is a single gene mutation, a single metabolic thing that's isolated. We can identify cell-specific mechanisms and work our way through that process. However, that style of thinking has very poor explanatory power for chronic disease persistence. So the toy example is somebody that has post-traumatic epilepsy. The cause is unequivocally being hit in the head by whatever it was, but the hit in the head is not persistent. The hit in the head is not the thing that's leading to the disease persistence because it's gone away. So this very linear process of the cause is the thing that we have to go after starts to fail at this level. It has little ability to explain disease change over time. So many diseases fluctuate, they progress. But if the cause is the cause, unless you are hypothesizing that the cause is somehow getting worse over time, it really doesn't explain why we see variability in disease outcomes. So it has this inability to explain variability. And as such, there's a very limited value for prevention and long-term care because it's not obvious what you go after from a therapeutic perspective. So let's go back to thinking about the pathophysiology of Alzheimer's disease. The thing that's been pursued heavily is this pathological protein aggregation. But all of these other things, synaptic things, proteostasis, cytoskeletal abnormalities, mitochondrial things, DNA things, inflammation, neuronal cell death, are all contributing to the outcomes of Alzheimer's disease. And in fact, it's broader than that, because if you look at epilepsy and if you look at autism spectrum disorders, you'll find a very similar range of things associated as pathophysiological processes. So now we have multiple mechanisms, and there isn't a single leading mechanism. In Alzheimer's disease, the pathological protein aggregation was deemed to be the leading mechanism, and in fact, the consequences of removing those protein aggregations hasn't been amazing. There are no linear relationships between the mechanisms. You can't say because pathological protein aggregation has gone up, that is in some linear way impacted synaptic function, which in some linear way is altering DNA defects, which in some linear way is altering inflammation. So it's very hard to draw that line. And therefore, it's better to think of this as all mechanisms contributing to the cognitive phenotype. And in this frame, it's much easier to think about clinical variability as some much more complex function of the proportion of mechanism involved. So the outcome depends on how much pathological protein aggregation with how much altered energy homeostasis, etc. And the predominant effects change over time. So early on, you might mostly be seeing pathological protein aggregations, but at the end, you're almost certainly seeing much more neuronal cell death, which are related to each other, but again, not in a linear way. And again, in this circumstance, we've now got many things that we could be targeting for treatment and prevention. And we have, in fact, attempted to target many of those things independently, one at a time, to improve outcomes. And none of those things none of those treatments have had very positive effects for any of the disorders I've shown you there. So what I've left you with at this point is a whole bunch of problems and no solution to the problem. So what I want to try and convince you of is that considering the brain as a complex adaptive system, which it unequivocally is, we can then build an overarching formulation, and inside that formulation, we'll be able to think of new ways of thinking about disease and potentially new treatments for disease. So an obvious question is, what is a complex adaptive system? So these are protean. They are absolutely everywhere. Many, many things that we observe in the world are complex adaptive systems. And all of them involve very similar things. So all of them involve many components that are dynamically interacting, giving rise to hierarchical levels that exhibit, each level exhibiting common behaviors. And fortunately, this generalizes across systems, scales, and disciplines, which means that we can capture techniques from one discipline and apply them to other disciplines. So it's a nice broad thing. So what this would suggest in neuroscience, for example, is you've got a whole bunch of genes at this bottom level, and those interact to produce different cell types or neuronal types in different brain regions, which form together to form microcircuits, which join together to form a macrocircuit, and you end up with an emergent behavior. So in this frame, it's much harder to say this gene is responsible for some outcome without thinking about all of these things in between. So what we're really talking about is emergent behavior. So complex adaptive systems show this emergent behavior, and it cannot be simply inferred from the behavior of the components. So when we are asked to define a mechanism of disease, in the reductionist frame, you're going back to the gene or the protein or the receptor, and that is deemed to hold a lot of explanatory power. I'm arguing that that type of mechanism holds very little explanatory power, and in fact, a mechanism that we should be thinking about as a much more generic mechanism would be self-organization. So this is a very interesting idea, which describes the spontaneous emergence of global order, structure, or functionality, which is from local interactions amongst the components of the system without any control or planning. So you can see these sorts of organized phenomena in physical systems. So if you heat oil, then these little vortices form. There's no oil particle or oil molecule that's the lead oil molecule telling all the other ones what to do. So you can see it in a physical system. However, in biological systems, you also see it, and they are functional. So this is a termite mound, and these are functional in that they enable airflow through them, which means when the termites are in them in the middle of the hot Australian or the African sun, they are kept cool and they don't die. However, there's no single king termite. There's no termite that is telling all of the other termites what to do and drawing plans and building it. This is entirely a function of how the termites interact with each other and with the environment and with the weather. And what you get is this amazing, self-organized, very functional thing. And this happens broadly. The reason we get a Dow Jones Industrial Average is because investors are interacting with each other and driving each other's behaviors. You see it in ecologies. So if you want to understand which animals are living in Yellowstone and at what proportion, you need to understand who eats who and who lives near the river and who lives inside the trees, and they're all interacting with each other. Murmurations of birds is the same thing. So these are starlings, and this is a meta-murmuration where the starlings make a murmuration that looks like a bird. And this is also functional. Predator birds are much less likely to target starlings when they're inside a murmuration than when they're not inside a murmuration. And all of us that drive know that for reasons we don't understand sometimes, traffic stops and goes, and it's a function of how drivers are interacting with each other. So the question then becomes, is self-organization something we need to understand? And can we leverage the ability of any complex adaptive system to self-organize as a function of its environment? Can we ultimately change environments in a way that moves a complex adaptive system to produce a behavior that we want? So this idea was nicely summarized by Peter Dodds, who is a complexity scientist at the University of Vermont, who said there's no love in a carbon atom, no hurricane in a water molecule, and no financial collapse in a dollar bill. And in a traditional sense, what we look at in medicine is the carbon atom, the water molecule, and the dollar bill. And what I want to look at is the love, the hurricane, and the financial collapse. So I think this is how I want to conceptualize brain disease now. And I've simplified this enormously. So all the things that I've called mechanism and etiology are things like genes, structure, inflammation. I would now say all of these things are perturbations to some complex adaptive system. And those perturbations are the environment in which a complex adaptive system is functioning, and it is the emergence of that maladapted complex adaptive system, which is the thing that looks like the phenotype. So instead of necessarily going after the gene or the structure or the inflammation or any of those other things, we can say what's inside the box, and is that per se targetable in order to change the adaptation, make it more physiologically adapted in order to generate a more normal phenotype. So what's in the box is a whole bunch of networks. So you can think of this at the level of genes. So this is a gene expression network in Alzheimer's disease with upregulated and downregulated genes interacting with each other. A layer up, you could say, has a microcircuit with pyramidal neurons, which are sending information, interacting with interneurons, which modify how that firing happens, which are also associated and integrated and interacting with blood vessels and astrocytes, etc., etc. And you can go up a level to say what's happening at the level of the whole brain. And we can say there are networks of EEG abnormality, networks of functional MRI, and then networks of physical structural connections between brain regions, as seen here with DTI. So where I now stand, having said all of that, is I want to find somewhere inside this box that I can start to evaluate systems. So this is how we've been thinking about it. This was a paper we published a while ago really relating to epilepsy, where we're arguing that there are interactions of subcellular activity which are related to microcircuit neuronal activities with single neuron or multi-neuron recordings, and then regional pieces of the brain and how it works and how the whole brain works and what emerges as a phenotype. This is the emergence direction. However, there's also a feedback direction. So sensory information that's coming into the brain is understood at the level of the whole brain, regional brain activity, how microcircuits fire, and those microcircuits are influencing subcellular activity as well. So this is a thing that's in beautiful balance in physiology and is disrupted in disease. So as much as I'd love to understand that whole thing all at once, that's kind of not how it works. So you pick a level, and the level we've picked up to now really has been the microcircuit neuronal activity level and relating that to behavior and phenotypes. We are increasingly interested in what happens in the other direction and are starting to do subcellular activity experiments. And even though I don't have some of those, I'll show you some data from other groups that have looked in that direction to evaluate what sort of impacts we might get there. All right, so we are at the level of microcircuit neuronal activity, which means we need to be able to measure the microcircuit. So this is a summary of our approach. So what we do is we build these electrodes, which is up there on the left. This one has 4 tetrodes, so 4 sets of 4 wires tied together, which we implant into the hippocampus, into the CA1 region of the hippocampus. So we're in amongst all of these interacting neurons, and we capture information from them in these awake behaving rodents. As they're moving around, we capture action potentials across cells. What we can then do computationally post hoc is, because of the geometry of the neurons and the tetrodes, we're able to isolate individual cells. We isolate individual neurons and how those action potentials are behaving in time. So what we end up with is a whole bunch of binary strings for each neuron, where if a neuron is not firing in time, that's a 0, and if it fires in time, it gets a 1. So you get strings of 0s and 1s across a number of neurons simultaneously whilst the rodent is doing what it does. Okay. When we get all of that, we have to really try and understand what it means. So that's a lot of zeros and ones. And what we do is analyze them within these various constructs. So we look at changes in firing rate related to sensory input, and that's classically done in the visual cortex. Because we're in the hippocampus, this would traditionally be a place cell, which are hippocampal neurons which fire when you're in certain parts of physical space. Me sitting here means that certain neurons inside my hippocampus are firing now, and if I move to another part of the room, different neurons will be firing, which allows me to know where I am in physical space. We look at how neurons fire when they fire. So when they do fire, they don't just fire randomly or persistently. There is modulation of how those neurons are firing. That's called timing coding. And then we're interested in how these neurons fire with respect to each other. And that's called population coding. So this is the framework that I'm going to talk about the results in. So let's think a little bit about rate coding in neurological disorders. So what we have here is rats that have got a hippocampal brain injury from status epilepticus. And I'm showing you place cells in the top circles. So what this is you're looking top down on an environment that a rat is walking around in. And when the rat's walking, and different cells. Okay, so this is an environment, and these are the 4 different cells that we recorded while the rat was walking around. So when the rat was here, cell number 1 fired. When they moved over here, cell number 2 fired. And when they moved here, cell number 4 fired, but cell number 2 didn't fire anymore. Okay, so I hope that's clear. And what you can get is measures of with what fidelity, how precisely a neuron is firing as it gets into space. And this is coding the space. And what you find is in hippocampally injured animals, you've got less precise coding going on. So that's with a structural brain abnormality. Those animals have lost neurons. An alternative epilepsy model is early life seizures. So in these animals, we induce 50 seizures, short seizures over the first 10 days of life. We then let them grow up and look at what they're like when they are adults. And again, those animals still have a persistent of a firing abnormality, which is really about fidelity. We can look at a model of Alzheimer's disease. So this is the TPP model. And again, what you see at the top of the wild types is some nice high fidelity place cells, and in the APP model, some less high fidelity place cells. You can do the same thing for Fragile X, and you get a very similar result. You can do the same thing for methylazoxymethanol, which is a developmental model of a malformation of brain disease, which gives you very similar abnormalities of rate coding. And then you can do it outside of the hippocampus. So these are outside of place, and this is the fidelity at which a neuron knows which lever is being pressed during a particular behavioral test called a delayed match to sample test. So I've put these up like this because I wanted to show you that this is a range of different disorders, all causing similar disruptions to the fidelity of place coding, which starts to build this idea that when networks break, or when hippocampal networks break, there's only a certain number of ways they can break and only a certain number of emergent phenomena that arise. One of the things that we recognized reasonably early is that there's a very tight relationship between rate and timing coding. So all of those cells that are showing very strong place fidelity also have very typically modulated cells in time. And I'll talk about the modulation in time now. But the reason I think this is important is it makes it somewhat easier for us to go after timing coding. And from a treatment perspective, it feels more tractable to go after timing than about rate, because the cells that are being incorporated into a rate code are distributed. It's very difficult to know which ones they are. It's very difficult to know how we might drive them. So in fact, we went the other way and said we're going to think about timing coding. All right, so what I want to do now is use a disorder called Fragile X syndrome as an exemplar of the sorts of things that we're doing and the sorts of things that we're going to do in the future, given the new grant that we recently got. So Fragile X syndrome is a very common monogenic cause of autism spectrum disorders, which means these children have deficits in social communication and restricted repetitive patterns of behavior that are very typical of autism. And in addition, people with Fragile X syndrome have learning and memory impairments, anxiety, executive function, which are hippocampal prefrontal cortex relationships, and they also have epilepsy, which I won't talk about much more today. So what we wanted to know is whether our animal model had behaviors which were consistent with what we would expect to see in people with Fragile X syndrome. So we know that it has construct validity because it's about the loss of the Fragile X gene, which is exactly what happens in humans. So we're looking here for face validity in these mice or rats. We have both of them. So for a sociability experiment, we have this task where a rodent is put into a middle chamber, and in the two outer chambers is either another rodent or an object in the first instance. And rats and mice are both very social creatures, so what you expect them to do is spend more time with a friend than with an object when you let them explore freely. And in fact, that's exactly what you see here when you analyze that as a discrimination index of how much time the animal is spending with one side or the other. Whoops. So you see that very obviously here in this is the rat model. When you compare it with an old friend with a new friend, in the rat we didn't see very much, but I'll show you later in the mouse we saw very similar things, that most rodents want to spend time with their new friend rather than with the old friend. This is a task of learning and memory where a rodent has to learn an escape target where they can get a reward as a function of what's all around in the room so that they can triangulate. So what you expect is for a mouse or a rat to get better at finding this target over days. What you find in the controls is exactly what you expect, that the time dramatically falls away, and by day 5 the animals know to go straight to the hole. In the Fragile X animals, it's much more variable, and in fact, they use a different strategy than a hippocampal strategy to solve this. They're also more anxious. So in this task, you put the animals in a dark place and you leave them there. Animals that are anxious will stay much longer in the dark space, which means they'll spend much less time out in the light. So the controls, after a while, get comfortable that there's nothing in the light area and will come out and explore. The Fragile X animals won't. So they've got face validity and construct validity. So it's a good model for us to now try and understand what's happening in these animals' hippocampal networks, and then subsequently try and work out if we can target those. So what we find when we look at this neural network dysfunction during exploration is we absolutely find abnormalities. So from a timing coding, we have mathematical methods that allow us to evaluate this modulation in time. And it's usually a modulation at about 120 milliseconds. So what you see is when a neuron fires, it immediately fires again, and then has a period where it doesn't, and then fires 120 milliseconds later, the rest 120 milliseconds later. So this is a way of quantifying that. And what you see is in both the hippocampus and the prefrontal cortex, it's abnormal in the Fragile X animals. We asked 2 questions from a population perspective. We asked, one is if a neuron fires and then another neuron fires, do they have very similar patterns or not? And in that circumstance, the answer again is in the controls. When 2 neurons are firing simultaneously, they have very similar time encoding patterns than in the Fragile X animals. And if you do that in time, irrespective of what the patterns look like, you find that there are very significant disruptions, again, in how information is being shared between these neurons, either from a hippocampus to a hippocampus to a prefrontal cortex, or between prefrontal cortex cells. So what we conclude from this is that there is a network adaptation to the lack of the Fragile X gene and the FMRP, which is the protein, which generates a network that when these animals are exploring, so all of these animals were just walking around in an open environment, that they have diminished time encoding, fragmented population coding, and less structured hippocampal firing in conjunction with excessively structured PFC firing. And when we test the animals in the behavioral tasks, which is not while we're doing the same EEG analysis, we find that there are behavioral disruptions in social communication, learning, and memory and anxiety. So this really suggests to us that this quality of background dynamics, how the neurons are behaving in this exploring space, is a very strong predictor of the ability of a network to then be able to engage. So while the animal's just exploring, its network is waiting, it's poised, and as soon as it needs to do a learning and memory task, that network needs to jump to being able to do a learning and memory task, or a sociability task, or an anxiety task. Okay, so now, well, okay, before we go to the next thing, what we really wanted to know is whether this generalizes across disorders. So it goes back to the thing I was saying about rate coding. Again, when we look across all of these animal models at the timing coding things, they all look very similar to each other. So what Mohammed Khalifi did, who's one of our postdocs, is just take data that we had already recorded from the MAM model and an Alzheimer's disease model and a Fragile X model. And he took this pre-existing data, which wasn't built within a single experiment. And what he did is looked at the recordings, sorted out the spikes, built co-firing networks, looked at graph features, and then really did a topological evaluation of functional networks. So this is what we get: multiple cells firing in time. And what we then do is build pairwise co-firing maps, which we can then build a weighted matrix about that, which leads to a fully connected, weighted, undirected graph, which is what we then use for our subsequent analysis. So the first thing we looked at was edge weight distribution. So how well two neurons were firing with respect to each other. And we saw abnormalities across all of the diseases, but they were different types of abnormalities. So these histograms of the edge weight distributions are not the same as each other. So this one looks like selective strengthening, this one looks like some weighting or some weakening, and this looks like edges drop out from the control to the MAM animals. So that didn't look particularly the same, but what we did is looked at convergent metrics. So we then did a whole bunch of standard topological measures from a graph perspective. And this is 9 of the 12 measures that we made. And what we see is in all 9, the abnormalities all move in the same direction. So there is some level of similarity. And when we built a classifier in which we trained the classifier on, for example, Fragile X and Alzheimer's disease, and then tested whether it could identify the MAM animals versus controls, it was very good. So if we build this on 2 of the models and test it on the 3rd model in its multiple ways of doing that, we're always able to characterize the 3rd model. So these 2 things together make us think that this is, again, a convergent network that we can go after. So how we think about this is Fragile X, Alzheimer's disease, MAM, all have disruptions to their network, which converge onto inefficient, poorly functioning networks, and that leads to the cognitive impairment that we're observing. So where we want to get to is being able to build a network that is better and more effective and more efficient by whatever intervention we choose to have. So the intervention that we've been testing is brain stimulation. So the way that we've been doing this is we put an electrode inside a structure called the medial septum or diagonal band of Broca, which projects to the hippocampus. It's a pacemaker structure that projects to the hippocampus and sends timed information to the hippocampus. And all that we're trying to do is find an input to the medial septum. That means that the timed information that gets to the hippocampus means that the hippocampus is self-organizes, reorganizes, given its input, to be much more effective at performing a behavior. So again, we're going back to the Fragile X model, and we'll think about whether this works. So this is a novel object recognition task, which is another memory task. The way this works is you put an animal in a box, and they habituate to the box. You take them out, 10 minutes later, put them in the box where there are two similar objects, and you let them explore. Because there's nothing similar about them, they explore them the same. Take them out the box, put them back in the box. An hour later, having changed one of the objects, and the animal will choose, a normal animal will choose the object that's changed. So in this experiment, we had animals that had no stimulation, which were controls and Fragile X animals. Animals that were stimulated, which controls at 35 hertz, a range of stimulation frequency in the Fragile X animals, and then a longer term experiment that we also did. So if you look at this discrimination discrimination index, when the objects are similar, all of the animals are the same. None of them are doing anything that looks abnormal. What we did with the stimulated animals is we took them out the box, we stimulated them for 30 minutes, stopped the stimulation, and put them into the new box. So controls do what you expect them to do now. They are more likely to spend time with a novel object. However, the Fragile X animals still don't when they are unstimulated. And when you stimulate them at 7.5 hertz, 20 hertz, or 75 hertz, you get no effect whatsoever. But if you stimulate them at 35 hertz, you get a normalization. This is a very dramatic effect. And remember, the animals are not being stimulated anymore. This was stimulation prior to the task. Interestingly, the same stimulation actually made control animals worse. So the way we interpret that is you need to set up the network to be as effective as it can be. And in fact, 35 hertz in control is disruptive. And then what we did was stimulated 35 hertz for 30 minutes a day for 10 days, test them 24 hours later. It still had a very positive effect. And with no further stimulation, it still had a very positive effect 7 days later. So we were encouraged by these results, but wanted to know whether it generalizes. So here's a string of other tests that show that this is a generalizable phenomenon. So this again is a sociability task with a 30-minute stimulation between tasks. What you see is the controls distinguish between an animal and an object. Fragile X much less likely to. Fragile X stimulated at 35 hertz, like normal. When you do a familiar versus a new animal without a second stimulation in here, so one stimulation back there, controls do it, Fragile X without stimulation are poor, Fragile X with stimulation are like normals. The memory task is the same, so this is over days. What you see as the controls and the Fragile X are basically following the same line. The unstimulated animals are behaving abnormally. Here's an anxiety task, so different to the last one. You put them in the lighted area. They don't like it, so go in the dark area. But when they're in the dark area they get a shock. So they then come out of the dark area and stay in the lighted area. There's no difference between these in terms of them going into the box. 24 hours later, you put them back in the lighted area and you take away the shock. The Fragile X animals go back in the box and they find one control wasn't too scared to go back in. None of the stimulated animals went back in because they remembered the anxiety they felt when they went inside the dark area of the box. So this 30-minute stimulation prior to doing a task seems to have a very positive effect on the animal's ability to do the subsequent task that we ask them to do. We haven't done the in vivo experiments yet, but what we did do was an in vitro experiment. So again, we did a 30-minute stimulation, sacrificed the animal, took the hippocampus, and did a short-term plasticity and a long-term plasticity experiment. So this is slice electrophysiology. We have a stimulation electrode in the Schaffer collateral, and we watch what happens to neurons when we are stimulating here, which is the downstream effect. When we looked at two different things, so we looked at synaptic efficiency. So what you do here is give a stimulation at a certain frequency, and you expect the brain or the synapses to facilitate at a certain level, and ultimately that will flatten out. At 20 hertz, there's no real difference. The Fragile X animals have got a reasonable efficiency if they are seeing 20 hertz information. If that hippocampus is seeing 30 hertz stimulation, the unstimulated animals now are significantly different to the controls and the stimulated Fragile X animals, and that's true again at 40 hertz. So we've done something to allow a subsequent piece of information to be better transmitted through the network with that 30 hertz, with that 35 hertz stimulation pre-sacrifice of the animal. We then did long-term plasticity experiments. So this is a marker of memory. So what you do is give a whole bunch of stimulations and then give a high frequency stimulation and then observe how the hippocampus responds subsequently to that. And in the Fragile X animals, they respond poorly. The controls are in the middle. And in fact, the Fragile X animals here overshoot. And even though I haven't got it on here, when we published this data, we had actually done 7.5 and 75 hertz as well. And they are somewhere in between the Fragile X and the stimulated animals. So there is some positive effect, but it's nowhere near as big as the 35 hertz effect. All right, so that's very exciting, and we want to go ahead and proceed to look at the in vivo electrophysiology, try and understand what we're doing to those networks with 35 hertz stimulation, and then ultimately think about whether that is translatable. But we also want to go down. So there's a question of how brain stimulation might affect cell metabolism. And it's clear that action potential dynamics play a crucial role in regulating cellular signaling with changes in gene expression, changes in inflammatory pathways, changes in oxidative stress, and reducing neuronal loss via trophic factors. So this is a model of Rett syndrome, which is a genetic disorder which is associated with autism and severe learning impairments. If you look at gene expression in models of that particular disorder, you see disruptions with upregulation and downregulations of genes. And if you stimulate the fornix, which has a very similar effect to stimulating the medial septum, and look at what happens subsequent to that, you get a 25% normalization, or just over 25% normalization of the gene transcriptome. So the genetic expression landscape is much more normal. And what you also need to know that if you do gene therapy and replace the gene in these models, you get the same effect. You get about a 25% normalization of that gene expression landscape. This is a model of pilocarpine. So this is a brain injury model. So if they give pilocarpine, the animals have a very long seizure, and then they have brain injury and inflammation subsequently. If you stimulate after the brain injury, what you see in the pilocarpine animals is they're way less likely to have apoptosis as measured by caspase-3, have altered or modulated inflammatory processes as observed by looking at IL-6, although TNF-alpha didn't show the thing. If you look at oxidative stress in a model of Parkinson's disease in mice and in monkeys, what you see is that brain stimulation recovers mitochondrial homeostasis. It's a very complicated study that I'm not going to try and explain the graphs to you, but what these graphs are showing is that oxidative stress is recovered. And in this model of epilepsy, looking at brain stimulation in a different structure, the medial prefrontal cortex, what you can see is animals that are stimulated have increased brain-derived neuronal function, brain-derived neurotropic factor, BDNF, which is a trophic thing which keeps cells alive. And nu-N just counts the number of neurons that are there. And what you can see is in stimulated animals, you're able to recover some of these biological processes. So this is a string of processes that I showed to you when I was showing you the picture of Alzheimer's disease. So where this is now taking us to is if we can find the right stimulation into these structures so that we can get improvement in brain functional architecture at the level of action potentials, we can potentially expect that several of those pathophysiological processes that we've been chasing can be recovered simultaneously. And our hypothesis, therefore, is that this has the potential to be disease modifying in a completely disease agnostic way. What it comes down to is understanding the neural dynamics, which I've shown you already can be similar across a string of different disorders. So if we can identify people with those sorts of abnormalities, irrespective of the underlying etiology, we may be able to have disease modifying treatment with brain stimulation. Okay, but a lot of people are a little bit queasy about deep brain stimulation. And therefore, we also want to think about whether there are non-invasive ways of modifying these sorts of networks. So a long time ago, we looked at the effect of environmental enrichment in the MAM model. So this is a model of animals with a malformation of cortical development who have cognitive and social impairments. And what we do is we either grow them up in an environment like this with friends and toys, or we grow them up in a standard laboratory environment. And what you can see, and this is Carl Jenks who did this experiment, is in a water maze experiment, which is like a Barnes maze experiment. Animals have to identify a space in a hidden environment. Happens to be in water here. But over time, they need to learn the task and learn the space, which they do. So in controls, if you enrich them, you do get some sort of effect, but they end up at the same place. In the MAM non-enriched animals, they are always poor. And in the MAM enriched animals, they end up at a much better place. So environmental enrichment is having a positive behavioral effect. And we can see that with sociability as well. And when we looked at this in single units after the environmental enrichment, what we showed was that the fidelity of place cells improved. The theta modulation, the timing coding improved. And in fact, this is the experiment where we identified that there was a linear relationship between those two things, or a somewhat linear relationship between those two things. This study got heavily criticized because MAM is not biologically a plausible model, or is not a clinically relevant model, I guess. And also that what we were comparing was a deprived environment against a normal environment rather than enrichment. And it's true that people that are seen with cognitive and behavioral impairments, we're already enriching their environments. So we're probably not going to get much more impact from doing that. So we went after a different type of non-invasive stimulation, which was the playing of sound. So what we did was take mice with Fragile X or controls and gave them audio stimulation of white noise for periods of time, usually 30 minutes or so, like we did with the deep brain stimulation, and then observed whether they did better after the non-invasive stimulation that we had to give over multiple days. And we split the Fragile X animals here into different ages. So these are immediately post-weaning, these are young adults, and these are older adults. And again, it's the same social experiment when you play sounds, and what you get when you compare the animal to the object is a positive effect of sound, not as dramatic as one that you got with deep brain stimulation, but again, with the discrimination index of a new versus familiar animal, again, we're getting positive behavioral effects in these animals. When you look at a memory task, again, a novel object recognition task, what you find here is if they're similar objects, they explore as you would expect. And then after playing noise to these animals, you get some positive effect at certain age groups. So there's an inkling of something that kind of makes us feel like if we can find the right non-invasive stimulation, whether that's noise or TMS or TDCS or TDACS, then we've got a chance of improving outcomes across a range of disorders in a non-invasive way. All right, so where we've got to is that the concepts of mechanisms of cognitive and behavioral impairments have evolved enormously. And I think in this systems frame is an evolution that's continuing to happen and is getting more and more appreciated. I think leveraging complex adaptive system principles and thinking about how we can modulate regulatory feedback mechanisms has a great potential for treating the complex neuropsychiatric disorders, but we really need to understand this in a complex adaptive systems frame and move away from that linear frame. Brain stimulation mechanisms really have potential for involving multiple aspects of a complex adaptive system hierarchy simultaneously, and we are hopeful that with time, non-invasive approaches may become appreciated. So if this is the sort of stuff you like listening to, then I did a podcast with the Toffler Trust, which really was called that, Rewiring Brain Research, Why Networks Matter as Much as Genes. So that would be another resource. And Matt Mahoney and I, who's a mathematician who I've done a lot of collaboration with, co-edited this book, A Complex Systems Approach to Epilepsy, which looks at complex systems and interactions from epigenetics all the way through to whole brain systems. And most importantly, science is a team sport. So even though I'm talking to you today, there are people that have been really critical to this. So my two main collaborators are Matt Mahoney, who's the mathematician who's now at Jackson Labs, Amanda Hernan, who is a basic neuroscientist who's also very computationally oriented. Mohammed Wadouz has done a lot of the fragile, he's done all of the Fragile X experiments. Mohammed Khalifi is the one who is now looking at whether we can combine those data. We have an in silico project, which is being managed by Patrick, in which we're trying to build an in silico hippocampus to do the same sort of thing. And I have a new postdoc called Zach, who is going to be taking on a chunk of these experiments. And of course, it also requires funding. So this is the group of people that have funded, or the groups of people and organizations that have funded us and are still funding us, some of them. And the one that we're very proud of is this Transformative Research Award, which really is a high risk, high reward mechanism, which is allowing us to take this somewhat unusual approach to understanding learning and memory. And with that, I am done.
Thank you so much, Rod. That was absolutely fantastic and really eye-opening on all these new ways of both understanding disease and maybe treating disease. Any questions from the audience? And we are a little, we have 1 minute.
Sorry, I overran a bit.
No problem.
I can stay for a few more minutes if you want me to. I don't have to run at one.
All right, well, I have a questions while we are waiting for the questions. So the stimulations that you showed, are these long-lasting effect, or maybe I've missed something? I was wondering, is it a one-time and you need to repeat the stimulations for the animals to get better? I mean, to have a long-lasting effect, or is it just a one-time and then it's done?
So
and they are doing better.
I don't think it's going to be one time and done. So we did one time and then immediately did the task, and that worked. We did 10 days of stimulation without any task, and then 24 hours tested and 7 days tested, and it was long lasting in that paradigm. So we have gone a bit down the line of how long will this last. In the end, we haven't really, well, I mean, we are changing the system, but there's probably still going to be some maladaptation going on in the absence of stimulation. So I wouldn't be surprised if over time this just goes back to being an abnormal brain again that we need to stimulate. So my guess would be we would have to have an implanted device and learn something about what stimulation needs to look like, and that would be a long-term therapy, like for Parkinson's disease.
And does it have the I mean, do you see this stimulation or environmental stimulation effect in ways of rebuilding neurons? Because here you're talking about a system, so everything, all the neurons are interacting. But would it be regenerating? I mean, would it help regenerating new neurons?
So I don't think it can do that. I think neurons that are dead are dead. What we want to be able to do is prevent more of them from dying and to normalize their cell biology. So the thing that we are hopeful for and excited by the possibility is that if we do brain stimulation, a lot of those cell biology mechanisms that are killing cells might normalize. You can see less amyloid, you can see less oxidative stress, different gene expressions happen. It becomes a much more healthy system in which neurons are less likely to die. I don't think we're going to make new neurons.
I see. Well, that's really fascinating. Any other questions?
How does age factor into this complex systems approach? So most of our data is collected in adults, so they've already had the developmental component. I think this is just highlighting the distance that we've got to go, that this is nice preliminary data and it's exciting, but we're a long way from fully understanding how a brain maladapts during development and whether we have to intervene earlier, how late we can intervene, how neurodegeneration is working. So at the moment, I just think I'm accounting for it by saying these are evolving, maladapting, dynamic systems. So I expect them to change in time. The much harder question is, at which point in time do you have to capture them, and what do you need to do at that point in time in order to recover them? We're at the very beginnings of those sorts of things.
All right. Well, if no other questions, guess we're going to adjourn. It's difficult to do, but thank you so much.
Thank you very much.
This was really very exciting.